Okay, looks like it is almost ready to go. I am posting a link to YouTube in case you would rather watch it on YouTube. And we can go ahead and get started. Okay, let me go ahead and share my screen. Okay, welcome everybody back. This is day 9 of May Institute on Computation and Statistics for Spectrometry and Proteomics. We are very happy to see that you are staying with us. I hope you are not too exhausted yet. We have some really interesting topics left for the next 3 days. Today, we are going to talk about statistical analysis in MS Stats, specifically for quantitative experiments with TMT labeling. So I hope that this will be of interest to you. If you happen to join us for the first time, let me just review the logistics. Please use the chat box at the bottom for things such as, hi, how are you? We can't hear. But for all questions related to science, please use Q&A, because then we can manage the questions easier, and we can also save the questions and post them on our docs document so that you can review the answers later on. Okay, so then we will go ahead and get started. So the plan for today is that first, we will have the introduction to the MS Stats ecosystem by Devon Koller. So Devon is a PhD student in the Curry College of Computer Sciences, and he is one of the lead developers of MS Stats. He has done a lot of work, in particular with MS Stats PTM, and also he is the lead developer of MS Stats Shiny. So we will hear more about this today. Then I will speak about the statistical methods specifically implemented in Mstats TMT, which is one of the packages in the Mstats ecosystem. If you're joining us for the first time, my name is Olga Vitek. I'm a faculty in the Curry College of Computer Sciences at Northeastern, and our lab is the main developer of Mstats. And let me right away introduce Ting Huang, who will then do some hands-on analysis talking about how we actually work with TMT-labeled experiments. So Ting holds a PhD in computer science from Northeastern University. And as part of her research, Ting has developed a lot of the statistical methods, which we will discuss today as part of Mstats TMT. Very recently, Ting graduated, and she is now a research scientist at SEER, continuing to work in quantitative mass spectrometry-based proteomics. So we're very excited to have Devon and Ting with us today. So let us go ahead and get started. So first, we will do the overview of the Mstats ecosystem. Thanks so much. So Devon, you can go ahead.
Awesome. Okay, hopefully everyone can see my screen. Yeah, like Olga mentioned, I'm going to go over the MS Stats ecosystem today, just go over what packages are available, how you can use them, and then their kind of current status and what we're developing and what you can expect in the future. So first, I'm just going to go over the team so you can kind of get a face to everybody who's developing everything. I've been developing some of the PTM work, Msnetslip and Shiny, like Olga mentioned. Mateus is now the lead developer of the base MS Stats. Melanie has developed some of the Galaxy implementations. And then Mina and Ting are both former members of the lab. Mina worked a lot on the base MS Stats, and like Olga mentioned, Ting worked a lot on the TMT side. So if you have any questions or post in the Google group or anything, it's probably going to be me or Matt or Olga that are really looking at it, just so you know who is actually going to be there to answer any questions that you might have. Okay, so I think Olga showed this slide a bit yesterday. This is kind of the high overview of all of the different packages and how they work. So I wanted to go through it real quick and just kind of show you what is going on, because it can be it can kind of be a lot. So at the top, you can see all of the different signal processing tools that we currently have converters for. So you can see like Skyline, Max Quant, Philosopher. We don't have converters for every package. So for example, our newer packages like Mstats, PTM, and Lip only have a couple of converters. So we are actively developing the converters, but Mstats and Mstats TMT should have most of these converters available for you. All of these converters are contained in this new package that we created called Mstats Convert. You don't actually need to interact with this package. It's more of a backend for all of the other Mstats packages. But we have it here just so you kind of know like how everything works. And if you're looking at the code, if you see Mstats Convert, it contains all the code for these converters. Mstats Convert is called by basically everything, but let's just focus on Mstats and Mstats TMT. Base Mstats is used for significance analysis of DDA, DIA, SRM. It includes, like we went over yesterday, it includes visualizations, quality control, sample size, statistical analysis, everything that we went over yesterday. Mstats TMT also uses Mstats as the backend, but it's specifically for tandem mass tag quantification. We have two UI tools for Mstats and Mstats TMT. Galaxy, which Melanie developed, and also Mstats Shiny, which we went over yesterday. So only Mstats and Mstats TMT are currently available in the UI tools, but Mstats PTM is also being integrated into Shiny right now. Mstats PTM uses some of the backend from Mstats, Mstats TMT, and Mstats Convert, so it's kind of built on everything. It supports statistical characterization of PTMs, and we can remove some of the confounding with the global protein if you have an unmodified run that we can use as well. Msnetslip is built off of Mstats PTM, so it can also remove some confounding as well. And then you'll see kind of not really with any arrows pointed to it, Mstats LODB, which is for estimation of limit of blank and limit of detection. It has some packages for calculation or functions for calculation and visualization, but it doesn't really rely on anything, so it's kind of there on the side. But it is on Bioconductor, so you can use it if you would like to. And lastly, on this slide, we have the public data repository Massive Quant. So we have integrated Mstats into Massive Quant, so you can see if you have like an experiment, if there's some sort of quantitative analysis, you can go in and see not only the R code for the entire workflow, but also kind of the different steps. So you can see like, okay, here is the Mstats required data format. Here's the quantification summarization files. And then here's our differential analysis files. So it's kind of just a way to tie everything together and to make everything reproducible and be able to give out the package and data to everybody. So that's a general overview of all of the different packages. Msnetslip is available. It's available on Bioconductor right now. And I'll kind of go through each package individually and show kind of their status. Msnetslip is our newest package, so I think it was just put on Bioconductor in last October, but it's still in active development, so we just released a kind of new update just a couple weeks ago for it. Okay, so first let's go through Mstats. This is a bit okay, so let's just start with the downloads. So we over the past year, we've seen 621 downloads per month for Mstats. The current version is version 4.41, and this includes some refactored code, which was actually implemented previously, but I just want to go over it because it did increase the speed and memory consumption of the package, or decrease the memory consumption, increase the speed. How we did this was replaced a lot of the tidyverse or dplyr functionality with base R and data table functionalities. And we also moved a lot of the core conversion functionalities to Mstats Convert, which helped us be able to just share those conversion functions between packages. Currently, we're developing some new functionality to analyze very large data sets that don't fit into memory. So we've been running into some experiments with maybe like 50 gigabyte plus data files that like basically you can't load into R. So Matthias has been working on trying to um, just a functionality where you don't actually have to load it all into memory and then process it, so that we can actually handle those big data files and do analysis on them. So that should be coming along shortly. It is available on Bioconductor and GitHub, and I've included these slides in the Word document, so you can click on them. There's links there for all of this stuff. Okay, so next we have MStats TMT. MStats TMT has 234 downloads per month over the past year. Its new version is version 2.4. And in this version, we include that refactored code. So MStats TMT is based on MStats. So it has MStats Hall refactored code to increase the speed. And it also has itself has been refactored. So it should be a lot faster in this version. In addition, Ting just released in the new version, a new model to account for time series designs. So time series designs was already built into MStats modeling, but now we can also do it in MStats TMT, which improves our variance estimation of those designs. And I think I shared before, but the way you actually specify what kind of design it is in your annotation file. So there's a specific way you need to set up the bioreplicates and everything. So I can share that kind of vignette again, so everyone can see exactly how to do that. Again, available on Bioconductor and GitHub, so you can install it either way. The development version is on GitHub, and the live version is on Bioconductor. Okay, so next is MStats PTM. MStats PTM is a newer package. It's gone through a couple different iterations. So over the past year, it's had 109 downloads per month. Version 2.0 is on Bioconductor now. Version 2.0 contains a couple different things. So previously, we had a package called MStats PTM TMT, TMT PTM, which included PTM data for, or PTM functionality for TMT experiments. But what we did is just combine that functionality with the base MStats PTM. So now that other package, PTM TMT, has been discontinued, and now everything's just in this core function, which includes functionality for both label free and TMT. So you'll see if you actually use the package, you'll just have to indicate whether your experiment's a TMT or label free experiment. We also replaced the backend with the refactored MStats code. So before it was kind of on its own, didn't have like integration with the rest of the family of packages. So now, because we replaced the backend with the MStats code, we improved the speed and reduced some of the memory usage. So the package should function a lot better as well. This package was developed with as a collaboration with Genentech. And we also have, it is available on Bioconductor and GitHub. On the development side for this package, I think I mentioned before, but we are actively working on integrating the package into MStats Shiny. One of the challenges with doing that is that the package, since it's pretty new, we don't have as many converters as we do for base MStats and MStats TMT. So we are actively trying to build more converters into the function. So right now, the main converters we have are for max quant and progenesis. And it's doubly hard because we also have to find converters for TMT and different types of experiments. So if you do have an experiment that we don't currently have a converter for, and you can actually share the experiment with us, we'd be happy to work on building a converter for you, and then we can actually include the converter in the package so that more people can process those types of experiments, which would be great. Okay, and the last package that I'm going to review here is Msnetslip. Like I mentioned before, Msnetslip is our newest package. It was just released last October. Over that time, we've seen about 50 downloads per month. We just released a new version, 1.2.1, which includes just some more plotting functionalities. It includes some more vignettes so you can see different types of analysis you could do. It's been developed in collaboration with the Peptides lab. And like I said with PTM, we're still in active development. We don't have as many converters as we do for the other ones. So if you do have some converter you'd like to see and you can share the data with us, please do, and we can build out a converter for it. I think the main converters are Skyline and maybe Progenesis. I'd have to look at the actual package. And it's also available on Bioconductor and GitHub, and the links are in the slides. Okay, so those are the main MSStats packages. There's also MSStats LOBD. I didn't go over it here. Like I said, it's not really integrated in the main workflow, but it's available on Bioconductor if you'd like to use it. Next, I'll go over MSStats Shiny, which we obviously did a lot of work with yesterday, but just to give you an idea kind of the reason it was developed and when you might actually use it versus some of the code functionalities and when it might be appropriate. So first off, it's available online at the link you can see there, msstatsshiny.com, or the actual code version is available on GitHub, so you can run it locally. I just want to say that the online version does have limited RAM space, so it's just on a server, so you might not have as much capacity to process large data sets as you do on your local machine. So if you do have if you have a small data set, the online version will work fine, but if you have something that's a little bit larger and you want to run a little bit faster, you probably would want to just run the app on your local machine. Right now, it includes functionality for MSStats and MSStats Tmt with PTM actively in development. And the general goal of this app, it was to increase the usability and reproducibility of the MSStats analysis pipeline. So to give people who maybe aren't as familiar with writing R code the ability to use the packages, and also to be able to have like a simple workflow that different people can use. We can save your parameters and then you can reprocess your data sets later. So it should be really make things easier to actually reproduce as well. Okay, so I wanted to give an overview of kind of when you would use Shiny versus using the code. So when would you use Shiny? The main ideas when you would use the Shiny application are to use the MSS ecosystem without writing code. So like I said before, maybe you aren't as familiar with R or aren't familiar with coding. You can use the Shiny app and run an MSStats analysis the exact same as someone who would be able to write the code. It's really good. Even if you did download it locally, it still is better for experimental data sets that use limited memory. So the app itself, just because it's a UI application and not code, generally takes up some RAM on itself before you even start processing the data set. So it does require you to have kind of an overhead RAM to begin with. And so it really does work better for smaller experiments that aren't going to just take up all the memory on your computer. With that being said, one thing that's included in the Shiny functionality is the ability to produce code for your workflow. So what you could do is take, maybe if you wanted to be able to generate the code that you could use for a larger experiment, you could take a subset of your data set, run it through the Shiny pipeline with all of the parameters that you would like to use. So we have a lot of information on what parameters to choose. Everything that you choose gets put into this code file, and then you can download it. And then all you need to do is replace the CSVs in the code with your larger data sets. And then you can run the exact same thing that you ran in Shiny, just using the code. And you don't have to write the code yourself. You just run it in RStudio in the console, and it will be able to run the larger data set. And one thing that's really cool about that is, like I mentioned with MSStats, we're actively developing the ability to run very large data sets that don't fit into memory with MSStats. So that might not be possible with Shiny, but if you have some really big data set, you could do what I explained, just like run a smaller data set, download the code, and then you could use that functionality for the really big data set with the code version. So it gives you a lot of flexibility over like how you actually will run the experiment, and producing the code helps with reproducibility. With that being said, when would you want to kind of use the code version instead of the application? So obviously the code version works best when you have to process large data sets that require access to more memory. It doesn't have that RAM overhead that you have to use just to even get into the just to start processing everything. Additionally, one thing I think the code lets you do that MSStats Shiny, the application doesn't let you do, is it increases your flexibility to do analysis and plots outside of MSStats functionalities. So what does that mean? Basically, with Shiny, you need to have a data set from one of our converters or already in MSStats format. You need to be able to put it into the app, and then you are limited to the exact functions that MSStats can use to process the data. So if maybe you want to do some like data like exploration on your own that MSStats doesn't include, like some plotting that MSStats doesn't include, like do some histograms or something like that, or do some like change your data set in any way, that all has to be done outside of the application. So if you're comfortable writing R code, the code does give you more flexibility to do all of these different analyses using different packages, different plots that the app doesn't allow you to do, because we are constrained to what we actually put into the application. So that's kind of just to give you an idea of what's available between or when you might want to use each of these two, the Shiny or the code version. So hopefully you have a better idea of like what different, so hopefully you have a better idea of when you would like to use each of them going forward in your own experiments. Lastly, just to show you where you might want to reach out to us if you do have any questions. We have a Google group, so you can see there. You can post any questions you have on any of the packages, if there's any bugs, or if like you have a question on how you might process a data set or something's not working right. So post there. Matt or I or Olga will be able to respond and give you guidance. Additionally, you could post an issue on the package GitHub, and the links are all in the slides that we've gone over previously. So that's the overview of MSStats ecosystem. If anyone has any questions, I could definitely answer them now.
Thank you so much, Devon. I see there's like a couple of questions related to error messages and things like that. So I think we can just answer them by typing. So then let me go ahead and share my screen, and we will go back to statistical methods, and we will talk about statistical methods specifically implemented in MSStats TMT. And maybe I will have also, I will touch upon a couple more topics just based on the questions that we received yesterday during the presentation. And after that, we will have a small break, and then Ting will lead the hands-on session on MSStats TMT and based on Shiny. Okay, so the first thing is there were several questions yesterday asking whether MSStats can fit ANOVA models, and also there were no questions, but usually people ask whether MSStats can fit lemma, and what are the differences between ANOVA and lemma, which are more general methods in bioconductor, and the statistical methods in MSStats. So I will talk about that, and then we'll talk about some details related to MSStats TMT. So first of all, analysis of variance. So ANOVA stands for analysis of variance, and essentially it is an extension of the t-test that we discussed yesterday. Let me just review that. So let's say we have a label-free experiment which has one feature per protein. And so here we have a replicates from the healthy. So this is a set of healthy subjects with these values of protein abundance. these are the subjects with the disease with these values of protein abundance. And so the question we wanted to ask was whether in the underlying population, not just in our subjects, but in the underlying populations, the mean protein abundance in the healthy is same or different from the mean protein abundance in the disease. And so for that, what we did, we specified a null hypothesis saying that, well, we do not know what the population means are, but we'll make an assumption. So this assumption is that there is no regulation, so there is no change in the population level in protein abundance between healthy and disease. The alternative is that there is a change. And so to find some evidence against the null hypothesis, we built this signal to noise. So the signal is how much difference we actually observe from our data. And the noise is how much uncertainty we have associated with the two means. And so you remember that in the simple case, the signal was just the means in the two groups, and the noise was the standard error associated with those means. Remember, we talked about the central limits theorem, right? We talked about those S1 and S2, their variances in the population. So we have to divide by the sample size to reflect the uncertainty in the means. So this is what we discussed yesterday. Now, the problem, and in fact, there was a question about that also yesterday in one of the Q&A, what happens if we have low intensity data, or what happens when we have noisy data? And so usually what happens is two things. So either one of the two things. So either our data are very noisy. So if we have very broad spread of protein abundances, either because the protein is just highly variable naturally from one individual to another, or because our measurement technology adds a lot of noise, then the signal can be very small compared to the noise, and we do not have enough sensitivity. Even if there is an actual regulation, the noise in our data can be so high that we will not be able to detect the signal. So this is one thing that can happen. And the other thing is when we have a small sample size, then occasionally those signals tend to be exaggerated, because let's say if we only had 2 replicates in this group, we have replicate here, in the other group we have replicate here. So it appears that the signal is much larger than it should be, just because we haven't sampled enough of the population. So these are the problems, especially when the sample size is small. And so there are multiple techniques to handle this. Well, one technique obviously is increase the sample size, which is the easiest one, right? But there are also other techniques we can use to add stability to our signal to noise estimation. And analysis of variance is essentially an extension of this type of approach, which focuses on adding more groups to our experiment. And so here is an example. So this was a collaboration that we had a long time ago with clinicians who study cardiovascular disease. So now instead of 2 groups, we actually have 5 groups. Each group has its own population mean of protein abundance. There is absolutely no reason why every group has to have the same mean protein abundance in the population. However, what we can assume is that the variability underlying each group in the population is the same. So the spread around the mean is the same in each population. If we can assume that, then we can learn the amount of the variation in each population from all the groups. Even if we're interested in comparing these 2 groups, we can leverage the information about the variability from the other groups to essentially gain stability for our estimates of noise. So this is a technique related to the experimental design. So of course, first thing, just increasing the sample size is definitely a good idea, is definitely helpful. But another way to add stability is actually add more conditions. Now this comes with the price of the assumption that the variation is the same in every group. If it is not the same, then we need to do something more complex potentially. And so the question was whether MStats or Ms Tmt can feed this type of models. The answer is yes, but only when it is appropriate for the experimental design. So we will need to see whether the experiment contains more than 2 groups, we also need to see whether all of these observations in each group can be assumed independent. If they are not, such as in experiments with TMT, as we will discuss, then we need to do something else. So yes, we can fit ANOVA models, but only for the experimental designs when this is actually appropriate. And the good news is that MSStats does a lot of parsing in the backend to figure out which model is actually appropriate for each experimental design. But this is the use of the analysis of variance potentially. So, and this is just some details on that, right? So what we can, so we can, when I say that we learn the variance from each group, what we do, we actually say, okay, let us combine the estimate of variability, assume that it's the same variability in every group, and we have this pooled average of variation, rather than having these 2 separate estimates of variation. Okay, so this was the ANOVA. Now, the second thing that we can do is to add empirical Bayes moderation. So let me just review here a little bit, right? So if we assume the same variance between the groups, we just have a combined estimate of variance, and we have a combined estimate of sample size, and now we just have one. So using additional stability for the noise, so for the denominator that we have here, can also be done with additional procedures. And this is what Limma is actually implementing. So analysis of variance says, okay, let me borrow the information across multiple groups within the same protein. Limma now goes one step further and says, let me also borrow the information on the variance, not just within protein, but across multiple proteins. So now it goes one step further in that. So first of all, it will assume, just like analysis of variance assumes, that we have a common variance between all the conditions for each protein. So now this is how we have our signal to noise ratio. And same problem as before, if the sample size is small, this standard deviation in each condition, so this spread here, may be unstable just because we don't have enough data. And so Limma says, well, okay, but we have more than one protein. We have potentially thousands of proteins in our experiment. Can we somehow borrow the information across them? Well, one very kind of drastic way of borrowing the information is to say every single protein has the same variance. And this is way too restrictive because, of course, we know that every protein is not regulated in an identical manner. So some proteins are more variable, other proteins are less variable. So assuming that every protein has the same variance is really, really a strong assumption. What we can do, we can have somewhat of a middle ground, right? So we can assume that, okay, these variances of individual proteins, they all come from some kind of, common pattern, which is not a constant, but some kind of common pattern. And so technically, and this was developed quite a while ago for gene expression microarrays, but it's now also popular for proteomics. So what we do technically, just for technical reasons, we do not consider the variance of the proteins, but 1 over the variance. And we essentially plot a histogram of this quantities 1 over the variance over all proteins that we have, right? So we have many proteins, we assume common variance between the 2 groups, right? So we calculate this variance like this. And now we make a histogram of these variances over all the proteins. And then we can fit a particular distribution. This distribution is called inverse chi-square. And so essentially fitting a curve to this histogram. And this curve has 2 parameters. There is D0 and S0. And this S0, you can think of this as the average or consensus variance over all the proteins. So this would be the variance which we would use if we wanted to assume that every protein has the same variance in the data set. And then D0, you can think of this as a weight, is how common it is for all the proteins to have the same variance versus not. In particular, so if this histogram is very tight, so if it doesn't vary very much, then D0 will be very large. If the histogram is very broad, it means that from one protein to another, there is a lot of kind of differences in the variance. Then this D0 will be small. And so now there's actually quite a bit of theoretical argument, which essentially boils down to the following. Instead of using a protein-specific variance in this noise part, we use a weighted average, where we replace the variance with a weighted combination of protein-specific variance and the common constant variance between all the proteins. So before, each protein has its own variance, and it may be unstable if the sample size is small. Now this variance is stabilized because we have a weighted average between something which is calculated from the entire data set and something which is calculated only from the protein. How big the weights are, well, this is determined by this curve fit and by how flexible or widely spread this curve is. So to summarize, what lemma is doing is that it improves the stability of variance for comparisons such as two-group comparison or analysis of variance comparison. It is a good idea, it is helpful. What lemma does not do, it does not account essentially for differences in the signal. So it still assumes that all of the points that we have are independent, and it does not account for more complex structures of experimental design, which have some kind of similarities between observations, such as time course data, or paired designs, or repeated measurements, or mixtures if we have a multiplex experiment. And so lemma is really helping with the stabilization, but for many experiments in proteomics, especially in multiplex proteomics, the signal part of lemma is actually not quite adapted to what we would need. And so what MStats is doing, it is kind of using the same ideas of stabilizing the variance denominator, but it models separately the signal specifically to the needs of quantitative proteomic experiments. And because the whole point is to improve the stability of the variance, if in fact your experiment has a large sample size, this type of approach, it is called empirical Bayes moderation. So this type of approach makes no difference anymore, because what will happen is that if you have a large sample size, your protein specific variance will dominate, and essentially there is almost no difference between the two approaches. In practice, our experience is that if you have more than 3 replicates, there is really no need for that. This works just fine. But if you have designs which are more complicated than just comparing several groups, then the noise part, the signal part, is not quite sufficient in what lemma is doing for the purpose of quantitative proteomics. So we implement this especially for TMT. We, I think, haven't done for the Bayes MStats because people now tend to have larger sample sizes in their experiments. So it's not as relevant. This is one of the older publications which shows essentially how the variance is stabilized. So this is the Volcano Plot, as we discussed, right? Like log fold change and p-value negative adjusted for multiple testing. So here we see that large fold changes may or may not be statistically significant just because of the instability in the estimation of fold changes, whereas here more systematically large fold changes are associated with small p-values because the transformation is more linear, right, from the fold change to the p-value, because the denominator is more similar across all the proteins. Okay, so I hope that Ting and Devon can answer questions as you type them, and if not, I'm happy to answer more questions after that during the break as well. Okay, so now we can talk about MStats TMT and what we actually do for a TMT specifically. So unlike label-free experiments where we have, this was an older cartoon, right, that we looked at yesterday, we have essentially one sample per mass spectrometry run, right? So let's say we have a protein digested and we have a particular peak for this protein for the healthy individual, and then in a separate run, we have a peak for the individual with the disease. And so when we attempt to compare the abundances of healthy versus disease, whatever artifacts happen in this run and whatever artifacts happen in this run, they add variation and uncertainty to the things that we are trying to compare. And so this means that we have, so on one hand, the samples are it's easier to do. The samples do not have to be labeled. There's no complex processing. On the other hand, the price is that we take longer, but also the artifacts, between run artifacts, impact our statistical analysis, impact our ability to detect changes potentially. So an alternative to this is label-based and isobaric labeling. And I hope we all know how it works, right? So that we digest proteins from different samples, and then we attach isobaric labeling tag, like a barcode, to each of the samples, and then we physically mix the samples together. So that now when we have a peak in this M over Z versus retention time space, this peak is actually a mixture from all of these sources that we had. And the quantification occurs at the level of the secondary or tandem mass spectra. So we have reporter ions, which characterize each of the original samples. So now, instead of looking at the intensities of these peaks, we're looking at the intensities of the reporter ions. And we have to summarize all this information as pertained to the protein to ask questions whether, for example, the healthy sample is different from the various disease samples. Now, this is fairly complicated because we are now changing the quantification from MS1 level to MS2 level. And on one hand, we now have multiple MS-MS spectra, and you can imagine that the reporter ions, which are part of a same MS-MS spectrum, they share some similarities. They're more similar to each other than reporter ions which belong to different MS-MS spectra. For example, if an MS-MS spectrum was sampled at close to the apex of the MS1 peak, these intensities will be higher. On the other hand, if the MS-MS spectrum was sampled closer to the tail of the peak, then these intensities will be lower. Simultaneously as a group, right? Just because there was less biological material during this time. So as a group, these reporter ions tend to kind of vary together, higher or lower. And so statistically, we have to account for these differences, right? Because they represent the technology and not the biology. At the same time, while MS-MS spectra, they are stochastic, meaning that we really don't quite have the control of how many MS-MS spectra we'll see for each peptide or each protein in every run. And so it is very possible that we will have different peptide ions in different runs. We may have several MS-MS spectra per peptide ion in one run, none in the other run. So there's a lot of uncertainty happens. Traditionally, multiplexing has been used, especially if we have enough reporter ions. These days, we can have quite a few, I think 14 or even 18, so that the entire experiment can be done in one mass spectrometry run. Then we don't need to worry about these issues. Msnetslip can do that, but really the strength of Msnetslip is working with experiments where we have more replicates or more conditions than what can fit into one multiplex. So we really focus on experiments with multiple mixtures. And this introduces enough statistical complexity that we had to have a separate package for that because it's quite different from label-free experiments. And there are issues such as how we account for different layers of uncertainty, but also how do we design optimal experiments when we do multiplexing. And so that's really what we focused on. So here is how Mstats looks at this data. You have seen a lot of profile plots yesterday for label-free. So here we have a more complicated plot because the experimental structure is more complicated. So here, let me just walk you through. Each panel is a mass spectrometry run. Some of these runs can be technical replicates of the same mixture. Some of these runs can be new mixtures, so that we mix different replicates in different mixtures. Now the vertical lines separate the channels. So these are the labels of the different channels. Y-axis is a log intensity. Again, we always work on the log scale of the peaks. And different colors here correspond to different peptide ions. And you can see a lot of things. So for example, here we have a lot of within peptide variation. some of this is noise, but hopefully some of this is biology, because we will have different biological samples labeled with different tags. And the objective of our study is actually detect these changes on average over all of the data that we have and over all the peptides and so on, to say, okay, well, these are the groups which are actually regulated. They are changing in abundance between conditions. But we see right away that there is also between mixture variation here. So for example, this is the same peptide ion. And exactly as I was saying, so here we actually have two MSMS spectra for this peptide ion. Here we have one, and the intensity shifted because we have probably this this mixture. In this mixture, this peptide ion was sampled at a different height of the peak. So this is the type of variation that we have to account for. But of course, a really big challenge for us is the inconsistency of quantification between mixtures. So here, for example, we have an empty channel. So this is a missing channel within a particular peptide ion. This doesn't happen very much, so happens into 1 or 2% of cases. Most likely, if we can sample a PSM, we can sample all the reporter ions within this PSM. However, what can happen very frequently is that the same peptide only appears in a few runs. For example, this red peptide ion is missing from many runs. This blue peptide ion is missing from many runs, and so on. And so this is the biggest challenge for us, because now not only the intensities shift between the mixtures, but also we don't even get to see the same ions, and they have different ionization efficiency and like different properties. So how can we integrate the information across multiple mixtures to still distinguish these artifacts which are due to data acquisition from the actual true biological signals? So this is the objective of Ms Tmt. And so now let me contrast the label-free versus label-based workflows from a statistical perspective. So here is how we were thinking about label-free experiments, right? So we have multiple runs. Each run corresponds to a subject, or we can even have multiple technical replicate runs corresponding to a subject. Multiple subjects are grouped by condition, and then we can have multiple features with maybe some missing values. So essentially the condition is the top grouping, top level for the grouping of our subjects. And then subject is the second level and run is the next level. Now, if we work with label-based experiments, things become more complicated because the top level of the grouping now is a mixture. And now within the mixture, we can have many ways to allocate conditions and replicates in one mixture versus in different mixtures. And we just have some kind of typical design patterns which people frequently use that we keep in mind when we think about these problems. So for example, here we have multiple mixtures. Let's say each mixture has multiple technical replicates. So that's already one thing, right? Now, within the mixture, we can have, and within one replicate, we can have multiple conditions. And within one condition, we can, and let's say we have a few conditions, let's say 2 or 3, then we have room to add multiple biological replicates from each condition within each plex, within each mixture. And so now what happens is that multiple biological replicates are similar to each other if they come from the same condition. But also all of these measurements are similar to each other if they come from one mixture and they come from one run. So in statistical language, it creates a correlation of observations which come from one mixture and one run. And we have to account for the correlation. And then again, we have features, but unlike in label-free, where we have missing values here and there, here we have missing values. If we think of a feature as a peptide ion, so then we have missing values much more extensively if a particular peptide ion was not at all observed in the experiment. And so now we have some observations here, but then these observations are missing in other mixtures and so on. So we need to account for all of that. So first of all, summarize the features and then make sure that the features are comparable, and then account for this orange and blue aspects of the experimental design to essentially build again the signal to noise ratio to understand what's the appropriate signal, what's the appropriate noise, and whether yes or no, the signal to noise is higher than as we expect by random chance. So that's really the work. And we will mirror the same workflow as in label free, but with modifications. So a really important aspect here is experimental design. Yesterday, I talked about the fact that we need to ask ourselves what is the biological question. So whether we want to compare protein abundances between conditions at the population level versus explore protein abundances versus discover biomarkers. Again, here we are interested in comparing protein abundances at the levels of the populations. But also now we have the technological aspects of experimental design. How do we allocate samples to mixtures, right, or to plexes? Is it better to put all conditions in one mix, or is it better to put all replicates of one condition in one mix and another condition in a separate mix? I'll tell you more about that, but let me give you the answer right away. Usually, you want to put representatives of every condition or every time point in one plex. So things that you're interested in comparing should be as close to each other as possible within the same level of this random source, which is a mixture. And then if you have another replicate, you create a second replicate mixture with other biological replicates of that, so that if one mixture is lost, you still have one mixture which has everything, all the conditions in it. So there's more complications, and something that we advocate for strongly is to have one reference channel, which is used for normalization. And in fact, it was already here. I hope you can see on your screens. One of the channels here is called norm. So this is a normalization channel. This channel has some biological material which is added in a constant concentration across all mixtures. Oftentimes it's some kind of pooled sample, and it's a good idea because then we are more sure that this channel has enough biological material that across multiple conditions the protein is abundant enough that this protein will be selected for fragmentation. But also this reference channel will be of a great help for us to make sure that all of these intensities can be corrected so that they are comparable between mass spectrometry runs. Sometimes there is a question whether we need to have more than one reference channel. We actually had experiments with 2 reference channels. We found that yes, it is helpful, however, not helpful enough to compensate for the loss of a channel that can be used for one biological replicate. So fundamentally, having an extra channel for an extra biological replicate is more useful. So one reference channel is enough. We have seen experiments without reference channels. It is not impossible to analyze them, but then essentially you have to make much more strong assumptions. So you make assumptions that on average, the experiments do not have as many missing values, that the average intensities are the same across runs. Reference channel essentially allows you to freeze you from these extra assumptions. You do not need to make these extra assumptions. And also reference channel allows you to work with unbalanced experiments where different mixtures can vary in number of replicates for each conditions, which happens often, especially if you work with clinical experiments where you cannot control exactly the number of samples. So reference channel really helps. One other thing that we see often with TMT is fractionation. So that also is part of the experimental design, and MS Stats can handle that. So that's also one aspect that needs to be included. Okay, so then we have data acquisition, and after that, data processing. So for data processing, as Devan and Mina mentioned previously, we have converters from quite a few tools, including MaxQuant and Proteome Discover and FragPipe, probably most relevant for the course. And the converters don't just format things, but they also do filtering. And so Tim can tell you more details about the filtering step, which is important. So for example, currently, actually all MS Stats workflows filter out shared peptides, meaning peptides which belong to multiple proteins, we tend to discard them. so that we use only the unique peptides for quantification. There's a threshold for PSM, so for peptide spectrum matches which have many missing reporter ions. It doesn't happen often, but if it happens, we filter them out. If we have multiple peptide ions, so multiple MS spectra with the same PSM, we keep the best. And there are some criteria for the best. This will be intensity, missing values, interference scores, and things like that. Combining fractions. So if a peptide is present in multiple fractions, fairly simple, we just keep the version in the fraction which has maximal mean intensity. So that's data processing. And so now the statistical analysis has two parts. One is well, actually several parts, normalization, summarization, the second normalization, and then modeling and inference. So I will walk you through that. And so again, this is the data structure that we have in mind. So the first step is the spectrum level normalization. So what can happen is that different labels, they have slightly different labeling efficiency, or sometimes things just happen, some kind of artifacts when we mix multiple samples together. And so here, this is how Ms Tmt visualizes this. So again, each panel is a mass spectrometry run. Each x-axis is the label, and y-axis is the log intensities of all the features in this run. So across all proteins, all peptides, everything. Because it's a DDA experiment, because typically we will have a fairly large number of proteins, we do assume that the median intensity should be constant across the runs. And so the first level of normalization just equalizes the medians across all the runs. This helps so that we kind of benchmark this on different data sets. It does help reduce some of these artifacts due to labeling efficiency and due to some issues with mixing. This is not the strongest normalization, but it helps to some extent. So this is what Ms Tmt is doing first. This is done across all the proteins. After that, everything we do is done separately for each protein. So this was all proteins together. Now the next step is separately for each protein. As we have seen, right, so in different runs, we will have different peptide ions and different PSM. This run has more PSM, this run has fewer, this run may have some others, so that the intensities between the runs are not comparable. That's okay, because currently we do the summarization separately for each run. If you just think about one run at a time, you can think of this as a label-free experiment, right? So we have different channels corresponding to different replicates. Here we have different features. And so what Ms Tmt is doing, we summarize this using the same robust procedure as we use in MS Stats in label-free experiments. So Tukey median polish. And occasionally you can choose to have imputation if you still have some missing values left, but usually there won't be many missing values left within a run at this stage. And so this would be the summary of this protein across all the features. So the summary of the protein separately in each run across all the features here. These summaries are not comparable between the runs yet because they're based on features with different identity and different intensity, strength, and so on. But now already the values are comparable within the run. So if you have one run, so this really is all you need to do. You would stop here. But the next level of normalization separately for each protein now makes sure that the intensities are comparable not only within the run, but also between the runs. And this is where we use the reference channel. So this is an example which actually has two reference channels just to kind of illustrate what's going on. So this purple circle is the reference channel. The red line is just some kind of average, right? And we see that here the purple points are on the average, here they are below, and here they are above. So what this protein level normalization would do, it would shift the purple reference channels so that they're all equalized. And whatever shift was applied in this run will be also applied to all the endogenous intensities at the same time. And the same thing here. Here it's downshifted, so the shift is applied to all the endogenous intensities. So the assumption is that whatever artifact happened to the reference channels, the same artifacts happen to all the endogenous channels of the same run. And so now that we applied this normalization, now these intensities, they are comparable, not only within the run, but also between the runs. And now we can take advantage of all the full structure of replication and of the full structure of experimental design to distinguish signal and noise. But for that, we need to model what is the signal and what the noise, right? And so these are the challenges for the statistical part. So represent the experimental design, accurately represent the peak intensities, estimate the sources of variation, and do hypothesis testing. So make some statements about population level analysis. So this is the structure of the data as we discussed. And just like for MS stats, this type of designs actually are somewhat known in statistical literature. They are called split plot designs. What is important here is that there is a restriction on randomization. We randomize the replicates within the mixture, then we randomize separately the mixtures, but the features we acquire, they are not randomized, right? They are all acquired simultaneously within each replicate. Because we have restriction for randomization, and this is really important, we have different sources of variation will be considered as noise, depending on what we're interested in comparing. And I will get to that in a minute. But for now, and this is now for those of you who have some statistical background, this will be a more complicated version of this linear mixed effects model that we wrote. So this is a generalization of the ANOVA model that we discussed in the very beginning. In a very special case, when you have one mixture and you have one feature, you can think of this as the ANOVA model, but it generalizes to multiple mixtures and so on. Importantly, it now has multiple sources of randomness. So we acknowledge the fact that the mixture is random, replicate run is random, in the sense that if we have to repeat the experiment, we will not have the same mixture again. It will not be identical, and we will not have the identical run again. And the important insight here is that even though the model is complicated, when we want to compare conditions, which is our question of interest, when we compare conditions, this comparison only depends on the summary of the features. It does not depend on the individual features. So we can do something similar to MS stats, where first, after normalizing and summarizing things, we kind of work on the summarized version of the data. And this allows us to specify a model which is simpler. Now, I'm not saying it's a simple model, but it's definitely simpler than what kind of original statistical version would look like. And the advantage of this approach now is that we can, again, generalize this to experiments which compare more than two groups, compare multiple groups. And of course, oftentimes we do TMT because we have even more complicated designs, such as time course, such as pair designs, or some hybrid designs have both multiple conditions and time points. And so these models allow us to actually fully express the complexity of the designs and capture the right source of variation to build the right signal to noise ratio. And just to kind of add to this, so remember lemma, right? Because oftentimes we have a few mixtures and variances can be unstable. Some of these noises that we estimate, we can actually use the same empirical base moderation where we borrow the information across all the proteins. So there is that aspect. So it is similar in lemma in that we stabilize the variance. It is different from lemma because we account for more complex structure of experiments when we start having TMT labeling. So just a little bit on the results, and Ting will tell you more about that. So what is important for us is several things. Well, of course, the performance is important, but what is also important is the number of testable proteins. So how many proteins can we actually analyze with this approach? And it turns out that because, lemma, but also sometimes people use HR or some commercial tools for this type of analysis, it turns out that when standard methods were developed for other purposes, like for gene expression, and we just borrow them for proteomics. They don't quite account for the patterns we have in proteomics. And it turns out that some patterns of missing values or some patterns of values that are absent, they cannot be handled with these implementations. In particular, AGR. Well, first of all, AGR is not even designed for quantitative intensities in the first place, but it's also not designed for certain patterns of missing values. And so this is a problem. So it reduces the number of proteins that can be tested in the first place. And then there are others. So for example, Limma in particular, in many cases, this empirical Bayes assumption is too strong. And so the variance component is too small. And so there are too many false positives. And proteome discover is kind of okay overall. But if we look at how we kind of actually estimate the false discovery rate, so how often we are wrong among the proteins which we claim are differentially abundant, or often we're correct. We can kind of see that this the Ms Tmt balances well the true positives and the false positives. So we'll tell you more about that. So I hope that Ting and Devon can answer some questions if you have. And again, I'm happy after I am done to answer more questions about this in writing. And so now the last part I just wanted to share with you is essentially a little bit more about complex designs. So if we have one mixture and two groups, and they're all in one TMT plex, it's really not that complicated. And when you don't need to worry about statistical sophistication. So we're really interested in experiments which are larger scale and have more complexity. And so the first aspect here is how to allocate the resources for future experiments. And remember, I was telling you yesterday that we often think in terms of sample size, how different experiments can be more or less efficient in terms of requiring more or less sample size. So this is the type of considerations that we have. Not all of this is implemented in MS Stats because this type of considerations need to happen before collecting the data. But let me tell you how people think about that, how statisticians think about this in general. So if we have, and this is a simplification, this is from an older paper that I wrote with Anne Oberg a long time ago, turns out to be very relevant currently. So if we, let's say we have 5 disease groups, and let's say we also have a multiplex experiment with 5 labels, just so that I can draw the picture easily, right? So then things are kind of clear. So we have one block, so one mixture, right? One plex, which will have one biological replicate from each disease group. With more replicates, so then we have another mixture, which will have one biological replicate from each group, and so on. So this is called a randomized complete block design. It is randomized because we randomly select biological replicates in each mixture. It's complete because each disease group is represented in each mixture, in each block. So these designs are very effective. Well, one, because if you lose a mixture, if your run fails, you still have other runs which are present. This is one thing, but the other thing is you can show when we talk about the signal to noise ratios that it minimizes the noise. And so it will also lead to the smallest number of replicates compared to some other allocations. So this is a very effective design. And I think it's probably not a news to most people here. We know that if we have multiple conditions, we want to allocate them all in the same mixture. But now what happens if we have only 4 labels? So what happens, and again, just for the sake of the argument, if we have more groups than labels, we cannot fit every group in one mixture. So then we have many ways of allocating groups to the mixtures. Well, you will probably say, hey, just let's have fewer groups. That's an option, right? But sometimes we really need to have more groups, and we cannot have enough labels. So there is a solution for that. And a solution for that is called a balanced incomplete block design. It is incomplete because now we cannot have every group presented in every plex. However, it is balanced because we can do this in a manner that is quite systematic. So you see that, for example, group 5 is only missing once from the mixture, and it's present in all the other mixtures. Group 4 is only missing once from another mixture, and it's present in every other mixture. Group 3 is only missing once. So essentially, every block is having every disease group missing once, and every disease group is missing once in every block. So this type of balanced design can be shown to be most effective, meaning, again, it minimizes the noise in our signal-to-noise ratio as compared to some other non-systematic allocations. And of course, we can do this even more extreme. What if we only have 2 labels? Imagine you're doing SILAC, for example, right? So then if we have multiple groups and we have multiple labels, we can show that the same kind of balanced incomplete idea can be extended to this pattern. Now, the problem is that we have now this pattern has to require more runs. For example, here, we need to actually have 5 mass spectrometry runs to make sure things are balanced. Here, we don't care. We can have 2, 3, 4 blocks, doesn't matter. Here, we really have to have 5 blocks present. Otherwise, things become unbalanced. So if we lose 1 run, it's more of a problem. Same thing here, we need to have 10 blocks in this case, so 10 runs to make sure that this balanced pattern is preserved. But again, if we can do that, then it means that our noise will be minimized compared to some other allocations of resources. In particular, the reference sample, for example, in SILAC, you can imagine spiking reference peptides for every endogenous run. So this is actually less effective from the statistical perspective than doing this type of balanced design. And so this is how statisticians will think about that. So remember this noise, the denominator in the signal-to-noise ratio. For all of these designs, we can actually calculate what the noise will be analytically. And so here is what happens if we can fit every condition in every block. Here's what happens if we can't fit it. So there's this coefficient, which is greater than 1. So it is larger than this, this part. However, we can calculate what it is. And so you can actually do the same type of sample size here. So this is what happens if you have a complete design, so enough labels. And these are the designs with 4 labels versus 2 labels. So we see that 4 labels is more efficient. It will minimize the noise as compared to 2 labels. So this is not implemented in MS Stats. This is something which requires statistical expertise that is specific to your experiment. But this is to say that there are ways to allocate the experiments in a more smart way. And so I guess the message here is that 1, talk to a statistician when designing these experiments. And 2, if you do have such an experiment, MS Stats actually can analyze it. So MS Stats can work with this incomplete designs or unbalanced designs and things like that. So there's really no limitation for MS Stats TMT in terms of how the samples are allocated to mixtures. So now I would like to just very briefly mention the designs with multiple sources of variation, because that's also what happens very often. And let me again highlight Ting. This was Ting's work as part of her PhD. So this is a fairly technical statistical work, but I will try to give you the intuition rather than technical details. So here is a simplified cartoon. So let's say we have 6 channels in a mixture. And so this will be an experiment with 2 mixtures. This is mixture number 1. This is mixture number 2. So we have 12 samples from 12 distinct people. Some of them are healthy and other disease. So condition 1 and condition 2. The experiment I discussed so far means that we have in one mixture 3 biological replicates from one condition, 3 biological replicates from another condition. This is mixture number 1. And then mixture number 2 will have 3 biological replicates from one condition and 3 biological replicates from another condition again. So that's what we discussed so far. So if you have only one mixture, you can do ANOVA here, there's no problem. If you have 2 mixtures, you need to account for the fact that these measurements are more similar to each other than these measurements. because they came from the same mixture, they share some artifacts. So this is what we can do. But oftentimes we are actually interested in measuring the same subject multiple times. Imagine, for example, that you have a person before treatment and after treatment, or you have a time course series, I don't know, some cell lines and they're stressed somehow, and then you measure the response to stress in time. So then we have yet another similarity or another correlation between the measurements, because measurements which come from the same person, they tend to be more similar to each other than measurements that come from different people. And so here is one example. So again, 12 samples, the same as before, but now in mixture one, we have 3 replicates from 3 time points of one person. And here we have 3 replicates at 3 time points from the second person. So this is mixture 1 and this is mixture 2. So now we have 2 layers of correlation, or 2 layers of randomness. One is the person, because all of these measurements come from one person. And this is another person. All the measurements will be similar to each other. But then the second layer of correlation comes from the mixture, because they were multiplexed together and they share some data acquisition artifacts. And those guys share some other data acquisition artifacts. And so this is where statistical modeling is important, because this can be analyzed with ANOVA. This cannot be analyzed with ANOVA. Here you need to have more complicated models which have multiple sources of variation. So technically in statistics, it means multiple variance components that need to be quantified from our data. And that's where things become fairly complicated. So I will not really go into the details of what these models look like. I'm happy to do this after the presentation, but I will just do 2 things just to provide you the intuition. One is, so maybe just one more thing I will say before that. If we want to compare time points, I think it's probably intuitive that comparing time points within the same person will give us less noise than comparing conditions between people, right? Because here we just want to see how things evolve in time, whereas here we want to see how things change between different people. Here the person is similar to itself, things just change in time. Here we have a difference due to conditions, but also difference due to different people. So this will be noisier comparison than this comparison. And if we model this correctly, then we will make sure that these comparisons are more sensitive. They have less noise in the denominator than these comparisons. And so now this is just some sense of how different models think about this. So this is for Tmt, including this empirical Bayes moderation. So this is the denominator in the signal to noise ratio when we compare times. This is the denominator when we compare subjects. And you see that, and each point is a protein. And so we see that for most proteins, comparisons between subjects are more noisy than comparisons in subjects. And it's quite intuitive. But now if you think about, so these 2, they're equivalent to ANOVA and t-test. These 2, they're lemma with different terms. It's interesting what happens. So these models effectively, they do not have the ability to distinguish the noise within subject and the noise between subjects. And what they do, they just apply some linear transformations essentially. So this is not the right way to distinguish these measurements. And so we think that this is really where the main advantage of Tmt is, where we have multiple mixtures, but also multiple layers of comparisons, for example, between subjects, within subjects, or between subjects. And here, just some, again, technical argument. Let me maybe start with this first. So this is more for those who are familiar with statistical methods. So if you look at comparisons between conditions versus comparisons between the times, if you look, well, the estimates of variability are different, but if you look specifically at how much data went into these comparisons in the degrees of freedom, we see that all the methods except Tmt, they use the same degrees of freedom. It's another indication that the within subject and between subject variation is not distinguished appropriately. And this is just to show that when the features are more variable, we will of course have larger p-values, and when the features are more consistent, we have smaller p-values. Okay, so as I said, it was somewhat technical, but I'm happy to clarify, and I'm sure Ting can also clarify a lot of things here. So very last thing, and this is something that we would like to share with you, and tomorrow there will be even more discussion on this topic, but I would like to give a preview already, is how can we benefit from the data which have been collected previously and analyzed previously. This effort in terms of data availability and reanalysis came out of the many method development efforts in our lab, because as we develop statistical methods, of course, we need to test them on many data sets. And data availability is a very big issue for us, because we want to work with data sets acquired on different workflows, on different instruments from different labs, to make sure that things are really stable and generalizable. And while doing that, we realized that being able to share and reuse data is really key for our community. And of course, we have repositories such as Pride and Massive, which store a lot of the information about the spectra. But what is missing is the annotations of the samples and the information required to actually do statistical analysis. So results of peak quantifications, results of normalizations, and so on. And tomorrow we'll hear a lot about that. There is a lot of efforts in the community to enable that. And so we have developed, in collaboration with Massive, we have developed one such way to store and reanalyze data. So this is called massive.quant, which is an extension to Massive. So this is the work by Mina, so she will be talking about this tomorrow. But essentially, Massive Quant allows us to store documented analysis of quantitative mass spectrometry-based experiments, not necessarily proteomics. So we now increasingly have mass spectrometry imaging and other data sets also in Massive Quant. The specific feature of Massive Quant is that it doesn't really prescribe any particular way to analyze the data. So even though we contributed to developing it, it's not at all, by no means, MS-stat-centric or anything like that. It is also not dependent on any particular data processing software. So essentially, you can submit your data, you can submit the results of data processing, you can submit the results of quantification, you can submit results of statistical analysis in with whatever tool you used. And the idea is that people can start at any intermediate step of data analysis. And for example, if you want to compare different statistical methods, you have the results of quantitative analysis stored and available for you. So this is very briefly the structure of the repository. So it has very general kind of bins. First is, okay, how the data were acquired. So the raw files, then the identification, so output of identification of spectra, quantification, how the peaks are found and quantified, and then statistical analysis. So you can use any of the tools. If you have executable code, you submit the data and the code so that it's entirely reproducible. If it's something that is point and click, such as commercial tool, you submit the input and the output and the tool version. And then the idea is that you take a data set and you can analyze it with some tool, and then you can analyze it with some other tool, and you can have any other package that you want. And these data sets have unique identifiers that you can track down all the steps, and you can start essentially with in any place. You can replace one identification tool with another. You can replace one statistical method with another. And it helps us understand, again, how much our choices of data processing and of statistical analysis impact the underlying biological conclusions. Because again, as I was saying yesterday, the mission of our lab is to make sure that we detect as much biology as possible, and we're as little biased by the artifacts of data processing as possible. So that's all I had to share for today. So let me once again thank everybody who contributed to this work. I would like to highlight Ting, who did a lot of work on the TMT part of things, Devon, who has been collaborating with Ting on the Shiny part that you will see in a minute. Of course, Mina, who has been behind the method developments, and Mateusz, who is a lot behind the general implementation and scalability. And also I would like to thank in particular the Genentech, who has been supporting quite a bit of this work. So I will stop sharing screen now. So I am happy to answer questions if you have any in writing, but I think it is a good time to take a break. So don't go too far away. Let's come back quarter to the hour. So 12:45 Eastern or quarter to the hour, wherever you live. And we will look at the analysis of TMT experiments now with the hands-on, with the Shiny, but also if you have questions about the code or error messages or anything like that, it's a really good time for that. So don't go too far away, and we will be back at 12:45. Okay, welcome back, everybody. Thank you for the questions. Please don't hesitate to send more questions in the chat to Ting or Devon or myself. So let's welcome back Ting. So Ting, it's your turn now. Thank you very much for joining us.
Hi, everyone. Let me see. It's okay here. Yeah, I'm Ting. Recently, last December, graduated from Olga's lab. I just attended my PhD holding ceremony last week. So thanks, Olga, for inviting me to give the tutorial again after I graduate. So today the tutorial will focus on, we will use the same, I think you already attended the MSDAS tutorial hands-on yesterday. We will continue using the same Shiny app, MSDAS Shiny, but now this time today we will focus on the TMT functions. So I just because I believe you have already installed the MS Shiny. I saw some issues asked in the Q&A about the installation. So if you have a problem today, I would recommend you just I will do it again. Like first download the whole MS Shiny repository again and reopen it. So like here, I will just first go to the Git repository and then download the whole repository to your local laptop. And then let's see here, where did I put them? Okay. So, and then I just move it to, okay. So that's the MS the zip folder you download. I will just create a folder called just whatever you would like. Here I would say MS TMT hands-on. Let's see. And then so okay, move the repository, the MS Shiny folder, you just download it and then unzip it. So here we need to first unzip it. I just delete the original zip file. So here is the MS Shiny folder. All the function you need is in this folder. And if you enter the folder, you will see a file called R project. Then just open the R project file. So it will open your R Studio. After you open your R Studio, actually it's quite straightforward to run the R Shiny. There are only like 2 command lines. If you couldn't remember it, it's already in the Google Doc. You can see it, just a library Shiny and the run app. So if you just copy it first, library Shiny, and then copy the run app function, and then run it. Sorry, I forgot the bracket. So if you just run it, then the Shiny MS Shiny will open. So that's I think you already seen this. You have already seen this page, welcome page, yesterday. So today we want to first run the MSStats PEP, but today we will focus on the DDA TMT part, which then you can select DDA TMT. Then now what we need to have a data to run the pipeline. So if you go to the, if you go to the Google Doc, there is the third step, download example and fragpep data. Today we will use a data set processed by fragpep. as an example. So if you click the link, and it will direct you to a folder which has the example data set for the practice today. And then I'm not sure if you've seen, okay. So then what you need here is, let me see. Okay, so then just download all the somehow, let me, okay. So I think if you copy the link, let me use another browser to open it. So in this browser, I don't have the user information. So all the sorry. So okay. If you suddenly I forgot where to download all the files, or I have to download it by one by one. So okay, yeah, you can select all the files and download it as a zip file. It's not big because I create a small data set for a very small data set for the practice. This should be already downloaded. Let me see. Okay, it's already in my download. Then just move it to the same folder. Or you can put whatever you want, but I would like to put them all in the same folder as the Msnets Shiny, and then unzip them. Again, just delete all the original zip file. So after you download it, in the folder, you can see actually there are two for the dataset practice. It requires two inputs. Same as Msnets. First, you will need the Msnets. We need the data file which contain the PSM peptide, the PSM data, and we also need annotation file which tell you what experimental design the dataset uses. And Ms Tmt will, based on the annotation file you given, you input the information you input. Ms Tmt will automatically decide, determine which design you are using and what's the proper model we should fit, like it should fit based on your design. I will talk about in more details later how should we prepare the annotation file. So currently, just assume you already have the annotation file. You are and then we will see how we continue the practice. In the second part of the hands-on, I will talk how to prepare the annotation using different data set examples. So, okay, now we have all the two data sets we want. Then let's go to the R Shiny. So let's can start the okay, so I'm not sure. So let's start the analysis. So first, with the type acquisition, you should select is DDA. And then for the DDA, what the data set what we provide actually it's processed by the MS Fragger. So you should select philosopher. If you are familiar with FragPep, there are several inside FragPep. There are several components. Like MS Fragger for database searching, and also the philosopher, which is the philosopher, which will give you the different outputs. So what we need from FragPep is the PSM file output by the philosopher. So that's why you see philosopher is equal to FragPep, because it's the part of the FragPep, and the output we need is the PSM file from the philosopher. So MSStats, FragPep, it's kind of different from other tools. For TMT, because we have in TMT, it may have multiple TMT mixtures. Just as you have seen in Olga's talk, we call it TMT mixture. It's because some other people may also call it an experiment or even a batch. But what we really mean here, a mixture is a mix of all the samples and label them with the different tags. And so you first label the samples with different TMT tags and mix them together as a single mixture, right? So then we call it a mixture. So in in FragPep, it actually create a folder. If you attend the course last week for the FragPep hands-on, you will know it create a folder for each mixture separately and generate a PSM file for each mixture. So if there are 10 mixtures in the FragPep in the dataset, then FragPep will generate 10 separate PSM files. So that's why actually what we upload, if it's a philosopher, the upload data here is a zip file, because we just zip all the 10, all the files from all the mixtures in a zipped file. It's easier for the uploading. So that's why if you see, if you for other files actually like a PD or max quant, you just need to upload the one single file. It use one single file to include all the PSM files from all the mixtures. But philosopher is kind of it's different from other tools. So now we first okay, in the I think I'm sorry, I forgot to mention the data set. So the data set, this data set we are using is make it small. So this data set actually is a public data set. What we it's here is a reference for the data set. Basically, it's sorry, I think it's a mistake. There's no, it's from the it's a CP tag data. Actually, the original data set is a large cohort data set. It studied the CCRCC cancer, and it's we call it a pair design. In the August talk, the second part is a time course data. So the original data set, it has 23 mixtures because it's a large cohort, and within each mixture you can see there are two groups. One is the cancer group, which is the CCRCC cancer group. Another one I label it by the blue color, and at the same time it also has each mixture also has a sample from healthy group, which is the control group, and I label it by the orange color. And another interesting here is why we call it pair design, because recently in the CPTAC large cohort data, they always use the paired sample, which means the cancer sample actually, and they will from the same patient, they will take a cancer sample, but they will also take adjacent normal tissue from the same patient. So then we have two samples from the same patient. One is the tumor, one is the normal tissue. Then they are paired, so that's why we call it such design a paired design. So here in the cell, the ID is the patient ID. You can see actually these two, such as these two, they are paired. They are from the same patient. So this type pair design is a special case of the time course design, because you can imagine it's a normal is a 10 point, and then it changed to the tumor is another 10 points. So it's special cause of 10 course, just so we have the 2 10 points. For this, but just for our practice, and each mixture in the original paper, it was fragmented into like 25 fractions. So it's a pretty large data set, and it was analyzed by FragPep. But for our practice, we only select a subset of the runs. With the like here, I only select 2 mixtures, mixture 1 and mixture 2, and also for each mixture only select 2 fractions. So totally the example data set, it had 4 runs, but the original data set it has like more than 400 runs. So that's how the data set, and this data set is processed by Msnetslip. And for this example data set, what's the file we got from Msnetslip FragPep? If you see, this is the data we got from FragPep. If you unzip it, you will see there are actually 2 files in the 2 CSV files in the folder. They are for so as I mentioned before, said before, philosopher from FragPep output a PSM file for each mixture separately. Then there are, since the example data set has 2 mixtures, so you can see 2 CSV file. And what is more so interesting here is why you see it's called Msnetslip CSV, because now actually we collaborate with FragPep. As Mina yesterday mentioned, Msnetslip and Ms Tmt, the whole package family, we require a certain format of the input. So different tools like PD, MaxQuant, OpenMS, or here is a FragPep, they have different output formats. So even for the same level, same type of data, like PSM level data, peptide level data, they all have different formats, different columns names. So that's why we collaborate with the tools developers. We see, okay, is it possible to give us to like from the tool output format, which is required by the format by the Msnetslip Tmt or Msnetslip, then the user don't need to do any manipulation of the data. They can directly use output from the tool and run Msnetslip and Tmt. So that's what happened here in FragPep. We collaborate with the FragPep and say, okay, they have a button, like if you run the FragPep, you will see there is a button, see output Msnetslip, like generate Msnetslip output. So that's how you get the file. So when you run FragPep and click the button, generate Msnetslip output, it will give you the Msnetslip format output. So here is the 2 files. Okay, then that's the how we get the output from philosopher. I will also explain, like for other tools, what the input file, data file we need. Then, okay, let's go to the our Shiny app. So here again, select TMT and the philosopher, and then just upload the zip file. You just download. Which is here, zip file. Open it, upload it, and then second file is annotation file, which tells the design of the data set. Sorry, okay, it's too many screens here. Okay, this actually is basically the design of the data set. What the annotation file tells just describe exactly the table, but in programming language, so the software can know the design. And then we need to upload the annotation file I already prepared. So it's the CSV Ms Tmt annotation and open it. So in the tools, usually they have under their additional options we have provided in the Shiny app. Because actually, if you run the command line of the code, the original function, it has more options about this step. But in the Shiny app, we just simplified to one option here. Is you can select which protein call ID, which column you would like to use as a protein ID. Because usually the tools, they provide multiple protein ID columns, such as like a protein column which has all the protein like multiple protein IDs as like protein groups as the protein ID, because they couldn't further distinguish the proteins in the protein group. And they may also have another column called like master or something like that, which they will select one representative protein from the protein groups. And sometimes the tool also provided a gene name as the protein gene name columns. So both of them, all of them can be used as a protein ID. Then you can decide which one you want to use as a protein ID in this here. So here we just use the default protein accession as a protein ID. Then let's just click on upload data. This step is it's running. I will just want this is a real data set. Even they're only for a smaller size, but it's a real data set. So this step, what it's running here is given the input data, given the annotation, the first they need to combine them, match them. Another thing is inside the function is because in particular, sorry, TMT in particular, it's very frequently TMT is followed by a fractionation, because the sample size after you pull all the mix all the sample together, the mixture is quite a big sample, large sample. Usually it's followed by a fractionation. So one important in this step, first step of Ms Tmt is to combine the fractions. What we do here, I think I saw participant ask a question in the Q&A is how do we combine the fractions? So ideal case, each fraction, different fraction, they have completely different sets of the peptides. That's like perfect case. But in practice, we know it's often seen that one peptide is identified like in multiple fractions. So which means for one same peptide, we have multiple measurements. Then the question is how to deal with such overlapped peptides. In Ms Tmt, what we do here, it's quite a straightforward way. If one peptide is identified in multiple fractions, we just select the fraction, the best fraction. So how do we define the best fraction is if the tool provides the interference score for TMT, we will select the fraction which has the best interference score. If the interference score is not available, and then we will check, okay, for each fraction, how many, because in each fraction there are multiple measurements, like multiple channels. Then we will check how many data points we have for each fraction for that particular peptide. So we will select the fraction which has the least missing values. If missing value is still same, then we will go to something like maximum mean value, which means we will calculate for each fraction, we will calculate a mean intensity across all the channels and select the fraction which has the best, which has the largest mean intensity. That's how we do the combine the fraction. And then after we select for the overlapped peptides, we just select the best fraction and then we combine all the fractions. So inside this step, there are also something inside this step, there also do some filter steps, like for the tools, if there are Q value, and then it will apply a Q value filtering, like the fracpep here. we will it will apply actually default QI identification score filter. And if there is also option, but this option actually, that's why we see in the command line you have more options, because you can also select do you want to use only unique value, or do you want to use unique peptides, or do you want to include the shared peptides? That's all the option in this step. So, okay, here is the summary of similar like MSStats. After you it's done, and then here is a summary of the data set. So as you see here, the first data summary of the experimental design. As I said, we have 2 mixtures here, and each mixture has 2 fractionations, and totally they are 4 runs. So all the information are correct. Technical replicates is MS run, and I will show you later in more details. But basically, sometimes we will run the same TMT mixture multiple times through the mass spec. Generate a technical replicated MS run. So in this case, we don't have such MS technical replicate. So it's just the one, which means we don't have the multiple technical replicates. And then for the data set, it only has cancer and healthy group. So it's 2 groups. So totally there are non biological replicates from the 2 groups. So just assume you already have the experimental design, and then the data summary of the data set, it only has totally has the because we only select actually 2 fraction of the data set, so it's just a like 6,000 proteins around that, and then like 17,000 peptides. Feature here we mean just precursor, like around 20,000 precursors, and this is the number of the peptides per protein. So when some proteins has a large number of peptides around like 59, and then is the range of the intensities. So this is the first several row of the out. After this step is after combine the 2 input files. That's the files final output it looks like. So see, it has first the protein information, peptide information, and charge information. So also it has the mixture information, channel information. The last is what it's the sample information, like what's the condition, normal, tumor, and what's the patient ID here, like bioreplicate is the patient ID. So, and last column is just the intensity, it's the original scale. Okay, so it's a first, like actually it's a kind of a double check of the data set and your annotation is correct or not from this page. If you see everything seems okay here, no problem, then we can continue to the next step. So in the next step, it's from here, it actually is different from our MSStats. In TMT, there are 3 steps, mainly 3 steps. Olga already mentioned in the TMT TMT workflow. We have 2 steps. So first is spectral peptide level global normalization, and then we summarize the peptide level data to protein level. Here is the second step, and the third step is if there is reference channel, we will TMT will perform per protein, like local protein normalization. It means per protein normalization based on the reference channel. So here, I think we have seen the question like, what if we don't have a reference channel? Currently, if you don't have a reference channel, this third step is unable to perform to do that. So if you don't have a reference channel, you can MSI TMT will only apply the global normalization here. Actually, we have the idea, but it hasn't implemented in the MSDATS TMT code. But actually, you can, if you know some programming a little bit, and you can create a fake reference channel by yourself by like average all the samples in a mixture. That's another way if you don't have a reference channel. So here is one parameter here. It's a cutoff. So what does it mean? In MSDAT TMT, MSDAT TMT provide a different way for the different methods for the summarizing peptide data to protein data. Okay, there are, as you see, there are 4 different methods. Median is quite straightforward. It's just for each protein, select the median intensities from all the features, all the precursors. So log sum mean, because the median is we first perform log2 transformation to all the intensities and select the median one. Log sum is kind of different. It's first sum of the raw intensities and do a log2 on the summation. And then Tukey median polish is what also MSDATS is using. So it's like an iterative. It's also robust summarization, similar like median, but it's more robust as median because it's just a fit linear model. Here, the last method is MSDATS. What's the difference between, I think we also often get the question is, what's the difference between MSDATS and Tukey median polish? Because from yesterday's lecture, you know, MSDATS internally it uses Tukey median polish. The difference actually between them is the missing value imputation. So because in MSDAT TMT, we directly call the function from MSDATS. It will have another layer of missing value imputation. So basically, you can understand MSDATS as Tukey median, like median missing value imputation in MSDATS plus the Tukey median polish summarization. So in order to impute, so it directly called the missing value imputation from MSDATS. And from yesterday, you may know in MSDATS is we want to in MSDATS we only want to impute the intensities due to low abundance, like below the detection limit. So you have option to define what's the low intensities. What are the low intensities? So if you think the lowest 1%, here is like I think 0.1% intensities are low intensities, then you can just under MSDAT TMT, MSDATS will just impute those very low intensities. If you didn't apply the cutoff, it will just not impute the missing values here. But if you apply a cutoff, it will impute the missing value, like it will impute the very low values, like the very low tail intensities. So here I just didn't didn't apply the cutoff. Then let's run the protein summarization. I think this step is depends on it. It can take long. It depends on how many mixtures you have, how many TMT mixture you have. Because it's like for each mixture, the running time is similar. But if you have like many mixtures, a very large cohort, like 25 mixtures, then this step may take I think for each mixture it may take 2 or 3 minutes. If you have a very large one, it may take long. But for this data set, it only has 2 mixtures, so it's okay. Let's see. Okay. So for this case, since while we are waiting for the running for the protein summarization running. Another thing I think for each of the normalization, first one is the peptide level global normalization. Olga already mentioned is which is actually simple median normalization to all the different channels and different order channels and the order run. For such summarization here, okay, you can see is the first picture has already done, and we are running the second picture. For such global normalization, it has assumption is all the samples, the samples from all the channel, all the run, they have a similar amount of materials. That's the assumption, but sometimes actually it's the protein, like total protein abundance in the sample, they may not exactly so, but sometimes the assumption may be not valid. That's why I think for the two normalization, you need to be careful whether you want to apply them, such as like in some thermal experiments, which I like for some experiment, like in different sample, you apply different temperature. So like TPP experiments. That's for such different temperature, actually it will influence most of the abundance of most of proteins, which means you couldn't expect that all the samples they have similar protein abundance, because indeed the treatment change the abundance of most proteins. That's why I think for the first peptide level normalization, when you apply it, you need to make sure the assumption is solid, is valid. And for the local protein normalization here, because it's based on the reference channel, so I think we ask we have a question about let's see. Oh, it's already done, but I think let me answer them. Okay, for the local protein normalization, because it requires a reference channel, then we have a question like what type of reference channel we would like to we would recommend. We often have questions, do we really need a reference channel? I think for reference channel, there are two functions. Object first is can use for normalize the batch effect between different mixtures. Second is usually we recommend you to use a reference channel which has a pool of the peptides from all the samples. Like select a small proportion of the peptides from all the samples and pull them together and use it as a reference channel. Because for such case, the reference channel, it will have all the peptides from all the samples. It will actually also benefits the identification step of the peptide identification. So because when you do the database searching, when you do the peptide identification, you do it for all the samples in a mixture together. So if there's one sample, it has all the peptides from the mixture, then it's easier to identify all the, it will benefit the identification, make it easier to identify the peptides, I think reduce the missing values. That's why we recommend, if it's possible, like better to use a pooled sample from pull the reference sample. Okay, I think that's here it's done. The protein summarization step is done. I think another thing I would like to mention is if like you can understand it's a Shiny app is a front end, and then the RStudio is the back end. So what you saw in Shiny app, it's all running in the backend. So in the RStudio, you can see all the information. Because in the package itself, it will output several log information to tell you how the program, how the analysis is going. It is shown in the RStudio. Actually, you can see the information like each step is done. We start the protein summarization function, and the first run is done, and first mixture done, and second mixture is done. So after you run all the protein summarization, this panel is same as MSStats. You can output the different level summary, because in the data set, first you can download the data as a feature level data and the protein level data. Feature level data is just the peptide level data, and protein level data. Protein level is the normalized summarized data, but it's per run, actually it's per mixture, it's per run per channel. Here you can also download the summary of the protein abundance. What's the difference between the summary and the protein level data here is, as we mentioned before, you saw for this data set. Okay, there are multiple. But this data set actually does not have the such case. But sometimes if you have technical replicates, like you have the multiple, you have multiple technical replicates or technical replicate run of the same mixture, same sample. Then when we do the like downstream, if you want to do a clustering or you want to do other type of analysis, you only want the sample level summarization, which means you want to which means you want to combine all the technical replicates. So here it will give you the option to first output a sample level summarization. Let's see if we want a sample level summarization, and then that's the sample level summarization. Let's see. Okay. You see, it's a wide format. What we call wide format is each row is one feature, one protein, and the different columns are different samples. This is a very typical input to many other tools like clustering tools or classification methods. So see each columns one sample. We have totally in the data set here, we have, let's see, to run, I think we have 19 samples from different groups. So that's all the samples. If there's nothing here, means it's missing values. Okay, and then we have also can make it as long format. It's same information, just how you put them in the table, how are different. If it's long format, it's just a protein and a group subject. So it's a normal group and it's subject ID totally. And then it's a log intensity, it's after normalization. You can also output as a group level summarization, like it's so it will give you the mean intensities, the summarized intensities for each group. Then you can, if you calculate the difference between them, subtract them, they are the difference between the two groups for each protein. So, and if you want to do the other like a classification, clustering, or other other analysis, you can just download the table you want, do the following analysis. So another very important, very useful plot here is what we have in Msnetslip TMT. It provides two types of plots. First is the quality control. I think Mina also you also show it yesterday, but it's slightly different since for TMT. As you see, what quality control for all the proteins. Like you see here, each panel here is one is why you see only two panels. So each panel here is one mixture. Originally, we have two full runs, but in the first step, we combine the two fractions corresponding to the mixture. So there are only two mixtures here, mixture 1, mixture 2. Within each mixture, we have the samples from normal, we have the samples from tumor. Each box here is one channel. Y-axis here actually is the log2 feature intensity, log2 precursor intensity. What you may notice, the box here, the median of the box here are all equal. So what it shows here is the output after the first step, peptide level normalization. So the peptide level normalization, you may so as we said before, it's quite simple. It's equalize the median of the across channels, like equalize the median of the box. So this is actually another quality control, a double check of your whether the normalization really works. The first step normalization really works. All the plots we provide here is like two. I think we have two aims. First is it's provide you the plot to visualize, to confirm whether each step works as you expect. So this quality control plot will let you know whether the first step, peptide level normalization, really works or not. See, it's all equal, then we know it works. So we are fine. Then we have another type of plot, which is the profile plot. For profile plot, it will let you know whether the local protein normalization works. So because this local protein, the local protein normalization is applied for each protein separately. Then profile plot is also, you need to select what protein you want to see. We just select the Q615. You see, it's this one show you the original precursor peptide level input, peptide level data. Again, each panel here is one mixture, and then each dot, so like each dot here is each line here is one precursor. There are two precursors in for this protein, and then each dot here is one channel. So again, we have the data from normal sample. We have the data from tumor sample. But this is the peptide level data. How do we know whether the summary works, whether the local protein normalization works? Then you can click the button here, show the plot with summary. See, it will give you the plot with summary. So, okay, I think another thing I've got, okay, that's in the plot we you see, oh, they are almost like they are quite similar across different mixture. It confirm normalization works. Another thing, because in the figure actually we didn't include the normalization channel, because when we run the protein summarization, the last step filtering, there is a step that you can remove the normalization channel, because I think some users, they just don't want the normalization channel in their out protein table. You can select to remove it. But if you select to keep it, then under make the plots again, it will give you the normalization channel here, and then you can confirm whether the two normalization channel, the normalization channel from different mixtures are equal or not. They should equal, otherwise it means the local protein normalization failed. Okay. So if you have any question about the app, the Shiny app, or any figures or plots, just please feel free to ask in the Q&A. Okay, So now it's like we have the normalized protein data abundance, or we are good to for the next step, which is the statistical inference. You can statistical modeling and the inference. It can tell you whether the protein, a particular protein is differentially abundant between different conditions. This step is same as MSStats. So there are also different. So first, in order to perform the statistical inference, we need to first tell. Just imagine when you perform the test, you need to first know what you really want to test, right? There are like for this data set, it's quite straightforward. It's only two conditions, cancer and health control. So then we can just test. Okay, we want to compare cancer with control. That's okay. So it's quite straightforward. Then you can select just all the possible pairwise comparison. Here we only have the because we only two conditions, so it's just a normal versus tumor. But if you see, you can also like we also provide more customized options. Like here, compare all against the one. So you can set like compare all against the normal, and then it will. But because it's only two conditions, always give you the same results. But then now it's tumor versus normal. So then you can also customize like the pairwise comparisons. You can select first group is normal. We want first group is tumor, and second group is normal, or you can reverse the normal and tumor. So this works. This you need it when you have like multiple conditions, like when you have like different time points and different phenotypes, then you want to customize it. So the last one option is you can even like input what all the comparisons you want. So here for this data set, we just do is all the pairwise. And then for the group, and then for the next step is you want to, when you run, before you run the group comparison, there is empirical-based moderation. So you really, okay, here where you can select whether what's the empirical-based moderation. I think we also have the question is because lemma was lemma already what lemma actually internal lemma, what it used is the empirical-based moderation to moderate the standard error of the test of the estimate fall change. But just because lemma is very popular in genomics and proteomics, so many times you may see people just say lemma method. Actually, what they really mean is empirical-based moderation. You can, it's particularly useful and beneficial when the sample size is small. Because when the sample size is small, within group, like if you only have 2 or 3 samples per group, when you estimate the variance within group, the 2 or 3 samples may give you a very unstable estimation, either very pretty large or pretty small. So this will give you some, it will lead to false positives or then the empirical-based moderation is to moderate the standard error, the variance, to make it more stable for the small sample size. So since the data set is small sample size, we still want to apply the empirical-based moderation. But when your sample size is becoming larger and larger, actually this whether apply or not apply the moderation, it will give you very similar results. And then just start the analysis. So what time is it now? Okay, we still have a half hour. So while we are waiting for it's running, I will start to just explain how you see, if you go to RStudio, it will tell you how the analysis is going. See, it's small, it's the progress bar tell you how much it has finished. It has finished. Let's see. Okay. I think we have a question about the fractionation. Because we are not, we didn't really run the white lab experiments. I think it's just the same sample and separated into different small fractions based on. Actually, I not couldn't remember the correct word into different based on maybe HPLC. I'm sorry, I really know that, but just from my understanding, they just split the whole TMT mixture into different fractions, and ideally the different fraction they will have completely different set of peptides based on some criterion to separate the sample, and then for each fraction just run through the mass spec. So while we are waiting for the running, it's almost done, but I think we can go to the how to prepare the annotation file. So, okay, for the annotation file, in the annotation file, what we need is the 7 columns in the fraction in the annotation file. So first, what's the annotation is we want the information, sample information, and the run information for each run and each channel. So like the data set, it has 4 runs and 10 channel per run. So we need to fill in the information for the 40. So the first the data set should have the annotation should have 40 rows, and we need to fill in the information for each row. And just to see per each run, each channel. That's the first 2 column information, run information, and for each run, the channel ID. And then for each run and each channel, what's the information we need? First, we want the run information. We want to know the run is the mixture ID. So like multiple run, they may have same mixture ID if they are the technical replicated run of the mixture, or if they are just the fractions of the mixture, then they should have the same mixture ID. Also, if its run is a technical replicated mixture, then just give the technical replicate ID. So if the run they are just a fraction, so they are the fraction, multiple fraction, different fraction of the different mixture, same mixture, then they should have the different fraction ID. So I think in the next slides I will show you how to prepare for this specific example data set. And the second type of information we need is really the sample information, which is very important for our testing. Because in order to perform testing, we need to know what you want to test. That's where you tell the software, the tool, the sample phenotype information. So basically 2 information. One is the condition. So like here, it's from cancer control, or it's from cancer. Another information is a bioreplicate, which is, you can understand it's like a subject ID. Like the example data set, we have 2 samples from same patient, then the 2 samples should have the same bioreplicate ID because it's the unique, the subject ID. Why we want subject information is because we expect that the sample from the same patient, they should be similar like the sample from different patients. Then actually it's a build additional block of the design. So we want to know this subject ID information. So okay, then for the data set. Okay, for this specific example data set, first is we need to pull out the run and the channel information from the input data from the data file. What do we do here? So first, open the philosopher MS CSV file. You got it from the philosopher FactPep, and then look at the columns. We need to look at two columns. First is the spectral left column. Spectral file column. The run ID is just exactly includes unique values in the spectral file column. So the spectral file basically is the run ID of the experiment from the experiments. Then this run column just exactly have the same ID as the spectral file. And then the channel column is just exactly is the channel column names you see in the CSV file. So if there are 10 channels in the CSV file, just put 10 channel ID in the channel columns, which means this Ms Tmt, the software actually it works for no matter what, how many for different TMT reagents, such as no matter you have 10 channels, like 10 plex TMT, or you have like now it's 18 plex TMT, as long as this channel column in the annotation file is matching with the channel columns in the data file, then you are good to run to use the Ms Tmt software. So that's how you get the first two runs, columns, run and channel. Then after you have the run and the channel, for the next information, it's quite straightforward. So first of all, we emphasize again on the size of the annotation file, because you have two mixtures, two fractions, so there are totally 4 runs in the data set. So we need the information for each run, each channel. So there are totally 40 columns, 40 rows in the annotation file. And then we need to fill in the information for each row. First is the run first. Actually, the run 1 is the fraction 1 of mixture 1. So the run information is just extract from the spectral file column, and then the channel column is just exactly the same. Same as the channel column here. And then for each run and each channel, we need to fill in first is the mixture ID. Since all this the sample here is just from the fraction mixture 1 and the fraction 1 of mixture 1, then it's just put 1 for mixture column and 1 for fraction column. This data set, it does not have the technical replicate MS run, so we just put 1 or any single value you would like to put under the technical replicate mixture column. It just tells you whether this MS run is a technical replicate of some mixtures. And then the next column, last 2 columns is condition and the Bell replicate. I think we have a question. So I think we have a question about the purity in the philosopher file. Actually, we have a purity cutoff in the first step. I think it's the default is 0.6. That's the FragPep team recommended. Like, as you can understand it as the 1 minus interference score in PD or other tool. So purity means larger is better, but interference score is like smaller is better. It's like 1 minus. You can understand it's a 1 minus interference score, the opposite of the interference score. Yeah, I think we have because I come to it, I'm not sure, but I think maybe David can like we can answer the. So we have a question about will the annotated sequence some flecting on the annotated sequence cause any error? sometimes yes, because we will, because if there are some I couldn't remember, there are a special symbol. If you use it in the annotated sequence, it will cause error for the downstream analysis, because sometimes we also use some special symbol, just a character combine. Because we sometimes separate the annotated sequence based on a certain pattern. If you use the same pattern, if the character you use is exactly just a coincidence, by coincidence, it's the same pattern as what we use in the code, then it will cause error. I think we can double check it and answer you later. So okay, continue the annotation here. So for the fraction 1, you see it's just a mixture 1, fraction 1. And then for the fraction 2, it's also quite straightforward. It's just a mixture 1 and fraction 2. Because we don't have a technical replicated one, so I'll put 1 in. We just put a 1 everywhere for the technical replicated nature. So and then see, this is the run ID is different. It's like F02 is like a fraction 2 of the first mixture. And then same again, the 10 channels, and just fill in the corresponding mixture condition and replicate. Actually, because we know it's the same fraction from it's a multiple fraction from same mixture. So this fraction, they should have exactly same condition column and the Bell replicate column. So you can just copy it from here to here. If it's and then go to mixture 2. So again, like it's mixture 2 ID, now it's changed to 2. Fraction is a first fraction is fraction, then 1 and the fraction column. And the run ID is also different. See, it's like mixture 2 and fraction 1. And all the 10 channels fill in the condition and the Bell replicate. So go to the fraction 2 of the mixture 2, or just fraction mixture 2 fraction 2, and again here is the condition and the Bell replicates. Here another thing I would like to point out is first, condition is quite straightforward. Is if it's normal, just a normal sample, just put normal. If it's tumor, just put tumor. But the fraction, multiple fraction from same mixture, they should have exactly like a condition column. But there are 2 keywords in the condition column. One, we also have the question before, I think, is here. One is norm. You see NORM. Why norm means this sample here is a particularly reference sample. It should be used for normalization. So it's a keyword. And just be careful when you put the condition name here, do not use norm, because the algorithm, the code, the software will think they are normalization channels. So norm just tell the software which channel should be used for the normalization. And another keyword is called empty. So we also have I think I have the question is, so sometimes we just don't have enough samples to fill in we just don't have enough samples to fill in all the channels. And then sometimes we saw indeed, in particular for large scale studies, there are some channels we don't have a sample to fill in. I would say we saw many studies. What they do is they will, because you don't have enough biological samples, but you may have enough material to generate multiple replicates of the same sample. So sometimes I saw scientists, they just put the replicates of the replicates of existing other samples and put them in the channels to fill in the channel. And then this type of replicates can another confirmation about the batch effects, the variance between samples, or between within sample and between sample and between mixtures. Also, but for such case, actually, when we do the testing, usually we don't want to use this empty channel for testing. even we put some replicates in the channel, but we don't want to consider them in the testing. Then what you can do is just put empty under condition. Then the algorithm, the testing statistical inference function know we will not use the empty channel for the statistical testing. So the empty keyword here is just basically let the user just select which sample you don't want to go into the testing, the statistical inference. That's the two keywords in the normal condition column. And for the Bell replicate column, it's quite I think only one thing you want to notice is like here, there is one normal sample, there is a tumor sample, they are from same patient, so they should have the same patient ID, like Bell replicate ID. Okay, I think that's how we prepare the annotation file for MSStats for the MSStats TMT. I know it's like compared with MSStats, we have more columns, but it's because we also have more complicated designs. So we want to let in order to let the tool make a correct decision, we need to tell them the correct information here. Okay, then let's see. Okay, then let's go to the next step, the testing one. I think we have the, okay, it's already done. So you see it's already done here. And then, so this part will list the significant proteins. Again, similar like MSStats, it will tell there are columns, protein columns, and label column just tell you what the comparison you just perform. And the log2 file change is the log2 difference ratio, log2 ratio between the two comparison, normal and tumor. And standard error is just the variance of the log2 file change, estimate of log2 file change, and the associated degree of freedom. This is, I think, if you can just simplify it, it's kind of related to the number, the sample size to add for this protein. So if the protein has more data points, then it will have the large degree of freedom. And so I think most interesting is the adjusted p-value here. So you can see that's the adjusted p-value column. So okay, that's the output you have. That's the file it outputs. And sometimes you may want to, I think usually for the testing part, we will want to look at the volcano plot. So again, here it has a volcano plot. But there are two options. You can either save it as a PDF or just view it in the browser. Let's see, we just view the plot in browser. See, it's the red. There are three colors. Gray just means they are not significant. Blue means they are downregulated, which means they have higher abundance in tumor. Upregulated means they have higher abundance in normal. So this is the volcano plot. One thing you may need to be cautious, be careful here is when you view the plot in browser, it can only view for one single plot. So if there are multiple comparisons and you see select all here and view plot here, it will return error. So every time if you want to view it in browser, just select one. But since there is only one, so it's equal. All is just normal to tumor. Okay, we have the last 50 minutes. I think we also you can just save it as in PDF. As a high quality PDF, then you can use it for your own like manuscript. Okay, so I think here for TMT, we haven't, for TMT, we are working on the sample size part, but it hasn't been finished. So in TMT, you don't have the function for sample size estimation and power analysis. That's the so then statistical inference is the last step in TMT. But another very new function I think Devin just add is download the analysis code. That's actually quite as Devin mentioned, it's quite useful because sometimes you want to have more flexibility because in the code you have more options. You same function, but there are many parameters. In the Shiny app, we only show us a limited number of parameters you can select. So in the code, you can select more you have more options for the parameters. So but if you just don't know how not that familiar with R programming, way to do that, like if you're not very familiar with the Ms Tmt command code or R programming, so it's the Shiny app actually is a good way for you to generate an R script. So we already run all the workflow. Then for the next, then you can download the analysis code. Like here, I just download it and save it in the desktop. So if you go to the desktop, that's the code we just saved. So click the green button here. It will just save you generate R screen, which include all the steps you perform in R Shiny, in the Msnet Shiny. So then you can. It's also like a record of what you have. Because in the Shiny, the problem is you do what you can do several steps and back and forth, but you don't keep it's hard to keep a record of what you have already clicked. But it generates the code will just tell you what the function you have generated. So okay. There's something wrong. Okay, so that's the code. I'm not sure what happened here. It seems there is. Oh, okay, here. So that's the code you have already run. Like here is the first. If you go to the Shiny app, the first step, data uploading. Uploading here, upload data. It basically run the function called philosopher to Msnetslip Tmt. So this is the here you need to tell them the code, what's the path to your data, and then it know the data. I also need to tell the path to the to your input to your file, to your data file. So you need the path to your input file. You need the path to your annotation file. And then the first function you run in the Shiny app is just basically the converter, which is upload data page, run the converter function, philosopher to Msnetslip. And then okay, so and then the data here, and then the next step is the summarization step. Okay, it's a data processing, which is the protein summarization step. It basically run the second function, protein summarization. Okay, and then the next step is also in the protein summarization function. It has the plots. So the additional data process plots will just output the plots for for the proteins, and also for all the proteins, or you can select each protein separately. So the last one is the statistical inference part. Then it's run the function group comparison TMT, and also the group comparison plots just give you the volcano plot. So that's the 3 steps. For again, converter for upload data, and protein summarization and process plot is for the protein summarization, and the last one is for the it's just for the testing part. So then you can fill in, like insert the path to your data, the path to your a file, annotation file, then run the code by yourself on the command line. So I think that's all the okay. So I think we have a question about the I think we have a question about adjusted p-value. So there are mainly 2, I think, like 2 types of p-value adjustment. One is the family-wise adjustment, which is the Bufferoni one. So Bufferoni one, it's the most straightforward to understand how we adjust it. Just divided the alpha, the significant level, by the number of the comparisons you have. But the problem for Bufferoni is it's super conservative, right? So it will make you have very few significant proteins. So that's why we have another type of adjustment method is like FDR based, which is a Benjamin Harshberger one. Actually, in Msnetslip and Msnetslip, what we use is just the Benjamin Harshberger one. So I think compared with Bufferoni, we use Benjamin Harshberger as the p-value just multiple testing adjustment method as default in Msnetslip and Tmt. So I think we we have this last 7 or 6 minutes. We will just stop here. So let's see. Okay, did I miss anything? I think that's it. Just I think for the families, if you can just practice on the Shiny app by yourself. If you have any questions, you can ask in Q&A. Or you can raise your hand and we can answer it online. If you want to practice, there is also an example data set. It's from the proteome discover. Actually, it's an example data set included in the Shiny app. It's output from proteome discover. Then you can see, you can use it to practice, or you can continue looking at the philosopher data set as practice. I think we have a error in, okay. In Q&A there is an error. I think we saw this error before, but it should be already fixed. Please make sure that Vijaya, just make sure you download the latest version of the RMS that's Shiny, because we used to have this error, but we think we have already fixed it in the most recent version of MS that's Shiny. Okay. So yeah, I think, and in the file, let me just, okay, there's so many things. So in the Google Doc, there's also course material which just link to the PDF I just talk about. In the PDF file, you will see there are more examples about how to prepare the annotation file. So I only, due to time reason, I only describe the first one, the example data set we use. But in the example, in the PDF, in the slides, it has for another data sets, like for all different cases, like data set from the PD is like only one meter, the simplest one, and also 2 meter from max quant is like a fraction, also which has fractionation, and also like a PD, but it's 5 meters, it's a large data set. This data set has technical replicator run. So you can, if you have a technical replicator run, you can refer to this data set to see how the annotation file is prepared. And for the last one, is the last data set is actually master from the massive quant. It's massive. It has the mass UCLD team. They have a master for database searching. You can, if you have the data output from master, even if they don't have a specific converter for master, but you can also use Ms Tmt for the this output, because the master output is same as the PD, very similar like PD. So you can use the PD converter for the master. And then for each data set, it just I think for the last 3 minutes, I just will briefly mention for this data set, the design is very interesting design, and also common design now is you have both different phenotypes. Like here it's control and the virus, infected samples and control samples, and for each sample you have different time points. Like here is for each phenotype, there are 4 different time points. So for such data set, what's different from is when you input the annotation file, what you input here is a condition. For condition column, you just can input a combination of the phenotype group with 10 points. So it's the condition now is control 2, control 6, control 10, control 24, and the virus 2, virus 6, virus 10, and virus 24. That's the difference if you have such combination of like a mix of conditions. So for such a combination, it's a combination of groups and 10 points. That's how you prepare the annotation file for such design. But then the question is how you testing them. When you're testing them, it's I don't have the time to go over it, but it's like just because when you for the okay here for the statistical inference, you have a create customized non-paired comparison. So if there for such design, you will see all the condition here, like control 2, control 1, control 4, control 8, and the virus were 2, 4, 8, 24. And then you can just select what you want to compare here and give them a coefficient. So okay, I think that's all the materials for hands-on for today. So feel free to, if you have question or if you have some installation issue, actually we feel free to stay here longer. We may have shares, we can like directly talk in the Zoom and share screen to figure out what's the issue in the installation. Okay, I think then so okay, I think thanks everyone again for attending our hands-on for Ms Tmt also Ms Stats, and also I would like to thank Devin, which is the developer of the Shiny app. Because it's really not easy to, we have the original function, but yeah, I think I can share the, so it's not easy to develop. So it's not easy to develop the Shiny app. I think the problem is at the beginning, we really want to have an online Shiny app for which one, so that everyone do not need to install the Shiny app locally and run all the files, all the Shiny app online. But actually, some of you may already try the online version. But the problem is really it's as a academic software, where it's hard to find a server or support the server, have a server to support so many users to run the code, the Shiny app at the same time. So Devin, he really spent quite a lot of time on how to improve the user interface and how to make the user can access the Shiny app online at the same time. So I think if you have a question about the package itself, or if you have a question about Shiny app later after the class, please feel free to email us or ask a question in our Google group. I think for the other example data set, yeah, we have the files. I will upload the files to the same folder. I think it's, let me see, I can feel it. So, okay, I will share the other data sets in the same, in the, add them in the Google Drive folder so you can download it later. Okay, thank you everyone. Then I think for today's hands-on, it's already it's done, and then just welcome if you have other question. Welcome to ask in Q&A. I will also stay here for a little bit longer in case you have any question about installation and the software. Okay, I think we have a question here. Let me see how I can make you wait. Yes, I think you can. You can talk now if you.
Yeah, hi.
Hi.
Yeah, thank you so much. Yeah, it was a great presentation. I really appreciate that.
Oh, thank you.
Yeah, I'm running to run the Shiny app. Like it opens the browser page, but I'm not able to access any of the tools there.
Let me see if you would like. Is it possible to share your screen? I'm not sure.
Yeah, I'm not having the option. Let me see.
Oh,
yeah, I don't see the option to share my screen.
Oh, I also couldn't change. Oh.
Is there a way I can send you a picture of how it looks like? Or
so how about, so now let's see what's the best way to. Okay. I think I couldn't let you share. Oh, I know how to do it.
Okay.
Let me add you as a panelists so then you can share your screen. Okay. How about now? Yeah, I think now you can probably can share the screen.
Let me see. Yes.
Okay.
Yeah, can you see my screen?
Yes.
Yeah, you see that it's there, but I'm not able to access any of them.
Interesting. Can you go to the Art Studio?
Yep.
I want to see what happened. Oh, just probably just close the Shiny app and
Yeah, it just said it couldn't perform the error. Execution halted and it closed the Shiny app.
I can first close the current Shiny app.
You want me to close it and reopen it?
Yeah.
Okay. Done the jobs. Saving it. Let me close this too. Screen sharing has stopped. Okay. Share screen again.
Okay, I see some error in the Shiny app.
Mm-hmm. It's
Okay. So where is the Shiny folder?
Pardon me?
Can you go to the folder, Shiny app folder?
Yeah.
Oh, I didn't see the files, the MS-Shiny files, the folder where
This one?
is a folder? How much?
Yeah, this is the folder I downloaded.
Okay, can you open the MS-Shiny? Probably because of the work directory problem.
Yeah, she's
Yeah.
1-2 in the work directory.
If you just run the app, click the run app.
Okay, I'm hitting the run app.
I think it's because of the work directory problem.
Oh, the working directory is a problem?
Yeah.
Okay, then let me try not using this working directory, download it somewhere else and retry it.
It's weird. See, it's already also, there is an error here.
You see an error?
I still couldn't click to use the app, right? Currently.
It said it couldn't find few packages. I was just trying to see if I have everything installed. It looked like I had everything.
It looks like one of the packages isn't installed too.
The Bellmat, yes.
Yeah.
Which one is not installed?
Can you run the Bell Manager install? Bell Manager installed the LAN full. Just
Can I go to the file manager?
The BIOC manager install Msnetslip and biomatter, that line 4 of the RF script right there.
upper
I
left.
tried this one. It goes every time, but I tried this one. But it still didn't. I'm gonna give it a minute, then retry it.
No, it's let me run the upper left, the LAN 4 Bell Manager. Copy it. Remove the.
Oh, okay, it's a comment.
Yes. Uncomment and run it. No, just not run app. It just how to copy it and run it on your command.
So
Yes. Console. Go to the console. Yes, console and run it.
okay, it's running few more packages. Maybe it
think,
was not sufficient to.
Yeah, one package seems missing. The second one, the biomat one.
The biomarat. Okay. Let me finish.
Just no.
Oh, you don't want all?
Because it may take a long time to update.
I'm sorry. I was like,
Fine.
update everything.
You don't need to update it. Just no, it's fine.
Okay.
Usually, we haven't finished it.
Yeah, it hasn't finished. I'm waiting.
So usually, it will take a long time to
Oh, I'm
upload.
sorry.
Okay.
Yep, there it is.
Wait. Can you scroll up to see the whole message, all the message? I just want to make sure. In the package list, can you check whether the biomat RT is over there?
Packages.
Yes. Bio updated. Just update.
Should I click
yes.
update?
Yes, update. Oh
You
no,
didn't
sorry.
have an update, right?
No,
It's
sorry.
updated.
Yeah, cancel it. I just want to make sure you have the.
Mhm.
Can you try again? Run up again. Not sure. It's...
Let me comment it again so it doesn't
Yes,
rerun
yes.
it.
Save, yes, save.
No.
Still. It's hard. What's the problem? Let's see.
If you run the other RStudio, I think it should work now.
Let me try that.
So like that other, yeah, like you have two RStudios open. So just
Oh.
run the other one.
Probably first the session. Can you go to session?
Oh, it's opening.
Not sure. Oh, it
Oh,
works.
it worked.
Yeah,
Okay.
the other one is just in the wrong working directory. So
Oh, it's just retaining it.
If you
Okay.
want, yeah, if you want to run in the other one, the one which has the error,
Mhm.
you need to set a directory. Like the
I'll do
one,
the set directory and over. Yeah. Okay, that makes sense. I had too many folders open there. Maybe that's the thing. Thank you so much. I appreciate that.
okay.
Yeah.
Okay, David, I think seems no question. We can just leave the meeting and close the meeting.
What was that?
I think we can stop recording. It's a no day.
Okay.
How about I just close all the